My name is Jolanda Van Leeuwen. I'm an assistant professor at the Center for Integrative Genomics at the University of Lausanne. So a lot of the research in my lab at the moment is focused on understanding genetic suppression interactions, which are genetic interactions in which the effect of a deleterious mutation is overcome by a second mutation elsewhere in the genome. And so the idea is, if we can understand how this happens and identify these suppressors, we can potentially identify new drug targets that can overcome deleterious effects of disease mutation. So I think genetic interactions have been studied quite a bit, mainly in model organisms, quite a lot. But genetic suppression, I think, has been a little bit understudied, in that we really needed a technology that is available right now to be able to do this. So this involves whole genome sequencing, CRISPR-Cas9, genome-wide screening, that up until a few years ago was either too expensive or not possible at all. So we used both yeast and mammalian cells as model systems. I think the advantage of yeast is still the throughput and the precision, which, even though we have CRISPR-Cas9 now, the throughput and precision of CRISPR screens are still not really at the level where we are with yeast. And so especially for our research, we want to identify thousands and thousands of interactions to try to understand how they happen. And this is still a little bit more feasible in yeast. But then of course there are things that human cells do that yeast cells cannot do. And so we also use human cells as a model system to try to understand interactions that are specific to the human context. So in the cancer dependency map, they're interested in understanding or they're identifying differences in phenotypes of a mutation across many different cell lines. And the research in my lab is also interested in understanding how the effect of a variant can differ across genetic backgrounds. And so there's many shared interests there. And so what we've been doing is we've been using some of the insights that we gained from our work to try to interpret DepMap data, but also the other way around, to use DepMap data to validate some of the findings that we make in different model systems or different contexts. And so I think we gain a lot from this huge systematic data set. I think the main challenges in the development of a cancer dependency map are that you always have to make choices of what you can study. Because even in a very large-scale project, you cannot study the effect of every single mutation in every single genetic background, in every possible condition and every cancer subtype. And so there always have to be choices that you try to make in the best possible way, but there's always going to be things that you miss because of that.